So welcome everybody to the July 2019 SNOC webinar, the Australian Inoculants Research Group. We worry about inoculant quality so you don't have to, by our own Jessica Rigg, and brought to you by the New South Wales Soils DPI Soils Unit. Now I'd like to introduce Dr. Jess Rigg, who's presenting today's webinar on the Australian Inoculants Research Group. Jess has worked with New South Wales DPI as part of the Australian Inoculant Research Group, or AIRG, since 2017. Microbial community analysis is where her research interests lie. Jess is particularly interested in how microbial communities can influence plant performance. Jess has worked with both beneficial microbes that promote plant growth, for example, rhizobia and arbuscular mycorrhizal fungi, and plant pathogens such as Phytophthora that cause disease. She investigated how microbial communities can influence the conservation of the critically endangered Wallamai pine. She's worked with the Royal Botanic Gardens in Sydney to study the impact of the root rot pathogen Phytophthora on alpine plant species. And she was involved in work on how amendments used in the current New South Wales DPI GRDC subsoil constraints project affected microbial communities. Now, I know you're dying to hear from Jess.
Okay, good morning. So it's really excellent to hear that some of you have heard about the green tick, and if you haven't, I'm going to be able to tell you about it today. So there's so many factors to consider when you're planting a legume, and so our group want to ensure that rhizobial inoculants that have our green tick are the best quality they can be, so that it's one thing farmers actually don't have to worry about is inoculant quality. So on the screen are some examples of some beautifully nodulated plants and some of the impacts that inoculation can have and the benefits it can have to legumes. So I wanted to start at the very basic beginning of how a nodule is formed. So when a seed germinates, as the roots grow, the plant releases flavonoids and other compounds, alerting the rhizobia to their presence in the soil. So the rhizobia respond to these plant signals by releasing their own compounds, and these are controlled by all sorts of nodulation genes. So as this molecular dialogue begins, the bacteria the root hair starts to form around the nodule. So this molecular dialogue is highly specific, and it's specific to the bacterial strain and the plant species and cultivar. So as this connection continues, the root hair curls around the rhizobia and an infection thread forms down into the root. A bacteroid or nodule forms. So next is a really example of a well-nodulated bean plant. So you can see a large number of nodules in the crown of the roots, and they're pink, meaning that they contain a plant-produced compound called legume, which is extremely important. It's essential to that, fixing nitrogen from the atmosphere into the soil. So rhizobia have a highly plastic genome. So basically, rhizobia can actually lose their ability to nodulate a host. So within a bacterial cell, there's the bacterial genome, which is a large circular chromosome. And if you'll think about this in terms of a computer, the chromosome is like the computer's operating system. It controls the day-to-day functioning of the bacteria. So bacteria also contain plasmids, small circular pieces of DNA. So these you can think about like software or apps. So basically, on your computer, you can delete a program or an app, and you lose the ability to perform the function that this app contains. So in the same way, nodulation genes are often included on plasmids of bacteria. And bacteria can actually lose these plasmids, or they can swap the information carried on these plasmids with other bacteria. So essentially, for some of our rhizobial strains, they may be the correct bacteria, but they can lose the ability to nodulate a host. So some of the forces acting on rhizobial evolution when they're out in the field include stress and competition. So basically, when an elite rhizobial strain is in the soil, it's competing with the whole soil microbial community. It's competing for space and resources. So when the rhizobia is free living in the soil. It's living as a saprotroph. So the genes that it contains to nodulate a host are expensive to maintain. So the costs to maintain the symbiosis genes are high, which means that they're more likely to lose these genes. So this is why, for our group, it's so important that we test our elite commercial strains annually to check that they still have this ability to nodulate the correct host, and we take steps to minimize genetic drift. So why do we even care about all of this? So this is quite complex to start. I'm going to talk about the benefit of inoculation. So there's many, many benefits to planting legumes and for them to be well nodulated. So you get increased plant biomass and forage quality, you can get increased grain yields and carryover for subsequent crops, and they can provide a disease break in crop rotations, plus many other benefits I haven't mentioned here. And this slide actually shows just a concentration gradient of high inoculant concentration to low, and you can see the benefit that inoculation gives. So in 2012, there was a back of envelope calculation that was done that said if all the area sown to legumes in Australia were fixing nitrogen optimally, we'd be saving $4 billion of nitrogen fertiliser annually in fixed nitrogen, which is a huge benchmark to live up to. So now I want to introduce my team, the Australian Inoculants Research Team. So we're a small soil microbial team based at Elizabeth MacArthur Ag Institute at Menangle. And so the role of our group is to basically ensure that Australian farmers have access to high quality microbial products. We facilitate a robust independent quality control system for microbial products. So we independently test products for inoculant manufacturers. We hope to increase consumer confidence in inoculant products, so by making sure they contain the right thing in the right numbers. And we also conduct soil microbial research. So our group curates the National Code of Practice and Green Tick Scheme, which I want to tell you about today. So basically there's two parts to this National Code of Practice. There's the protocols for selecting the elite commercial strains in Australia, and there is also the current numerical standards for legume inoculants. So under this program, there are 41 official commercial strains that have been developed over many, many years, and these cover over 100 legume species. And then within these species, there's many, many legume cultivars. So there's quite a broad reach of these commercial strains. So our National Code of Practice and Green Tick program are voluntary opt-in programs. So basically we have 4 major industry partners, which are listed there on the screen. And these partners choose to get their products independently tested by our service. And then when we independently test them, they are allowed to show the green tick logo on their packets. So if you see an inoculant bag that has the green tick logo, you know that it's been independently tested by us. And some of the things we're looking for is that the labeling on the package is correct. It shows what strain of bacteria is in the bag. We check that the correct rhizobial strain is there in present numbers and it meets in sufficient numbers, so it meets that quality standard. We also check that the product is free from contaminants. Contaminants in a batch may grow and outcompete the rhizobia in the packet, reducing rhizobial numbers and hence increasing the risk of low nodulation. So we make sure that there are low contaminants. And we also check that the product, after being manufactured, still nodulates a host. So when all of these things have been checked, we issue a green tick. So basically, if you purchase a product with a green tick, you can be assured that it's gone through that quality, those quality steps. So another key role of our group is to supply the commercial mother culture strains to industry. So we do annual mother culture testing. And as I mentioned, there's 41 commercial strains. So basically each year we check that the strains are pure, that the identity of the strain is correct, that strains can scale up for production. So basically they can grow to a large number of organisms, which is important for manufacture, and making sure those strains haven't lost those genetic essential genetic components required to form nodules. So an example on the photo here on the screen is of effectiveness testing. So basically, it's a subsample of an experiment where you can see plants that are inoculated are much larger and healthier and producing nodules than plants that weren't inoculated. So we also take these, we use a